All right, so here's the deal. We just learned how to do monohybrid crosses. And monohybrid crosses are crosses that involve using only one trait. Okay, so that's the key, mono, one. A dihybrid cross, on the other hand, is a cross that involves using two traits. So one of the things Mendel discovered was that traits sort independently. Just because I inherited blonde hair doesn't mean that I'm going to get blue eyes, because they're both, sorry, interruptions like usual. So just because I received one recessive trait doesn't mean I'm going to get the recessive trait of another. Just because I get the dominant of one doesn't mean I'm going to get the dominant of the other. So a dihybrid cross is going to be a cross where you can predict the offspring in terms of looking at two different traits at the same time. So we're going to look at an example. We're going to look at whether people have brown hair or blonde hair, and whether they have curly hair or straight hair. Okay? So we're going to follow the same process using our Punnett square. It's just much larger this time. Now, we're going to look at these genotypes in terms of parents. So we're looking at heterozygous for hair color and heterozygous for hair texture. Both parents are going to have the exact same genotype. So capital B, lowercase b, capital C, lowercase c. Now, when we look at gamete formation, right, males can create different combinations of gametes. So here's what I want you to think about. When I'm looking at the gametes, okay, if this is the male and those are the sperm, okay, and over here, this is mom and these are the eggs, okay, every single gamete has to receive two alleles. One for each trait. Okay? They have to get one for each trait. So that means in every single one of these lines, there has to be both a B and a C. Okay? Now, let's check this out. Different combinations. I'm pretty sure in math class you probably learned how to double bubble. Okay? If you don't know the term double bubble, then you at least know the term FOIL. So here, I can cross the capital B with the capital C, or I can cross the capital B with the lowercase c. So those are my first two combinations for gametes, for sperm, that the dad could make. So I'm going to write down capital B, capital C, capital B, and a lowercase c. Okay? Now, the ones that cross the bottom, now I'm going to use my second allele for hair color. And I'm going to cross the lowercase b with the capital C. And I'm going to cross the lowercase b with the lowercase c. So those are my four different gametes that the parent, or that the father could make. Now, because mom's genotype is exactly the same, that makes it a lot easier for me. So again, I use my foiling technique to figure out what are my different allele combinations. So I've got capital B, capital C, capital B, lowercase b, capital C, and lowercase b, lowercase c. So now I have to place the gametes into the chart. So it's time for us to look at the next page. Okay? This is an example of what a dihybrid cross would look like, okay, if we were looking at Mendel's peas. I figured you guys are sick and tired of talking about peas at this point, even though we're going to kind of be looking at corn in a little bit. So this is an example of the round versus yellow, heterozygous for both traits, cross. Notice how, instead of it being only 4 squares, now it's 16. So it's a much larger Punnett square to work with. All right, so this is the one that we're going to be working out of, and let me fix this. Let me get you squared away here. All right, hopefully you can see everything that I'm going to be writing up there. I don't want to cut anything off. All right, so first thing I have to do is I have to put my genotypes, my possible gametes, along each side, one for each parent. So let's take a look. I had capital B, capital C, capital B, lowercase c, lowercase b, capital C, and lowercase b, lowercase c. Now I have to do the same thing across the top. Okay, So now I can bring them together. So if you think about it, when you wrote genotypes on the monohybrid cross, you had the two letters that came together and you had your genotype. So when I read my genotypes, should I read BC, or should I be reading BB, I'm probably going to want to keep the alleles together for the genes that actually work for the same trait. So I want to keep my B's together. Sorry, interruptions. So in this case, my first box, my first offspring, is going to be homozygous dominant for hair color, homozygous dominant for hair texture, okay? So I'm going to keep working my way through the chart. I keep my B's together, and I keep my C's together. Now remember, we always want to write that dominant allele first, okay? I keep my B's together, dominant first, keep my C's together. B's together, keep my C's together, okay? So I'm going to continue going through and writing these out. See if you can keep up with me. Okay, so now I've got them all filled out, so let's take a look at it up a little bit closer. Whoa, sorry about that. So there's my results. Go ahead and pause it, finish yours, come back, take a look at it. So now, what's my next step? Well, my next step is to go through and identify my genotypes and my phenotypes. Okay, so now things are going to get a little bit more complicated. Okay. Let's use blue. In order to identify the genotype ratio, okay, I have to go through and identify what all of the different possible genes are. Okay, this is hard. So here's what I want you to do. You're going to list starting in the first square. Okay, so I've got capital B, capital B, capital C, capital C. How many of those do I actually have? There's only one, so I write down one, right? I'm going to go ahead and I'm going to cross that one off, so that way I don't count it a second time. So now I'm going to, it really doesn't matter what order you go in here, but I'm going to go through and I'm going to do this one next. So now I've got capital B, capital C, lowercase c. How many of those do I have? Hopefully you see two, one here and one there. So now I've got two of those. Capital B, lowercase b, capital C, capital C. How many of those do I have? Be careful, this one's a tricky one. There's the second one. So I've got two of those. All right, how about this guy right here? Big B, big C, little c. Heterozygous for both traits. How many of those do I have? You might see a pattern because there are 4 of those. Okay, I love how those go in a straight diagonal line. Because I'm kind of OCD, I have to do this one next. So I'm going to go ahead and cross that one off because I got big B, little c, little c. Huh. How many of those do I have? Just that one. I'm going to finish out this column. Big B, little c, little c. How many of those do I have? I see 2. This one right here, little b, big C, big C. Yeah, I only see 1 there. Little b, big C, little c. I see 2. And my last one, little b, little c, little c. There's only 1. Now, check this out. This is a huge genotype ratio. 1 to 2 to 2 to 4 to 1 to 2 to 1 to 2 to 1. Can I simplify that? No. There's no way to simplify that one. So now, let's check out the phenotype ratio. Because this is the one that is probably the most important. So I want you to focus, whenever we do dihybrid crosses, you're going to be focusing on the phenotypic ratio. Okay, this is the one that's very, very predictable, and this is the one that Mendel studied the most. So now I'm looking for hair color, hair texture. So I'm looking at, what do we got? Brown and curly, right? Curly was dominant, wasn't it? Let's go back and double check. Oh, we got to go back one more. Ooh, there I go again, pushing buttons. So curly was dominant, brown is dominant. So let's go back to our Punnett square. Okay, so how many of those brown and curlies do I have? Now watch how I do this one. Is this one brown and curly? Yep. So that one's brown and curly. Now I'm going to cross these off as I add them up. So I got one there. Is this one brown and curly? Yep. So I'm up to 3. Brown and curly, 5. Brown and curly, yep, that's 9. Is this one brown and curly? No. Is this one? Still no. Nope, nope, no. So I have 9 brown and curly. How many brown and straight are there? Okay, brown and straight, right? That's 3. Ooh, not brown and straight. So I only have 3. Okay, what is this one? Well, that one looks blonde and curly. How many of those do I have? That one and that one. So that one's 3 out of the 16. And then my last one that I've got left, the only one that actually gets recessive for hair color and recessive, whoop, silly equal sign, recessive for both is going to be 1. The phenotypic ratio for almost all dihybrid crosses, when the parents are both heterozygous for both traits, is going to be 9 to 3 to 3 to 1. In Mendel's studies, he did his experiments and he studied all of those thousands of plants, and he came out to almost, it was almost identical to the 9 to 3 to 3 to 1 ratio. Pretty cool stuff. So we're going to do some practice with those and take a look at a real life example of it.